Hello viewers, welcome to my channel Online Biology Tutorials. In today's live, we will be learning all the diagrams, how to draw each diagram that is given in the chapter, The Fundamental Unit of Life. This is chapter number 5 of class 9 Science NCERT that is followed in the CBSE board. So the very first diagram in the book is different types of cells. So in case in any of your assessment they are asking you to draw different types of cells, then I will help you out. In case they are asking about the cells present in the blood, so please do remember you will be drawing RBC like this, okay? Or the better one is this one. Okay, so I am deleting this one because the bottom one is better. And this one is telling you that RBCs, they are biconcave. Okay, so this cell is RBC. I can see that the screen is not clear. I hope it is clear now. So these are RBCs. In case you want to draw many, so you can draw like this. So RBCs are the biconcave cells in the blood. This shape is showing that they are biconcave, okay? So this is RBC in case you want to draw. Then the next cell that you can draw in your exam is you can draw a neuron. And to draw a neuron, you need to draw like this. I hope you know that neurons are the longest cells of your body. So in grade 9, if a question comes, how to draw a neuron, because there are different types of cells in the human body. So this shape is depicting that this is the long cell. So I'm writing it for you. This is the neuron. The topmost part here is known as cyton. And this part, in case you want to label, then you can. However, labeling is not required in class 9th. Only to draw the neuron is important. So till now I have drawn RBC. Now we have done the neuron. Let's come to the other cells. In case you want to draw a sperm, you can draw a cell like this, nucleus here. Every part has a special meaning if I am drawing like this, okay? So this is human sperm. In case you want to draw different types of cells, this is human sperm, right? And in case you want to draw an ovum, you can draw just a big cell, and this is the cytoplasm where I am marking the dot. So this is the ovum. So in fundamental unit of life, there are different types of cells being given to you. We have learned how to draw RBC, how to draw neuron. Now, this one was the third one, human sperm, and then the fourth one was ovum. Let's learn how to draw muscle cells as well. So in case you want to draw muscle cells, you can either draw a muscle like this, and this is the voluntary muscle that I am drawing right now, right? So this is the voluntary muscle. In case you want to draw different types of cells, voluntary muscle can be drawn like this. Hi Bunny, welcome to the live. And in case you want to draw the smooth muscle, which is there in our body, and it is involuntary, means it is not in our control, then it can be drawn like this. And if you want to draw cardiac muscles, then they can also be drawn like this. Cardiac muscles, right now I am drawing it for you. They are present in the heart. So in case the teacher is asking to draw a couple of different type of muscles, then these are the cardiac muscles, as the name itself is making clear that they are present in the heart. Right? So till now we have learned how to draw RBC, how to draw a neuron, how to draw human sperm, how to draw human ovum, and these three type of muscles: voluntary muscles, smooth muscles, and the cardiac muscles. So let's come on to the another part now, where we will be starting the organelles, how to draw each organelle. Okay, so first I'll teach you how to draw a plasma membrane, right? In case you are making an animal cell, then you can draw the plasma membrane like this. Okay, so plasma membrane is the outermost layer of the animal cell. Okay, so now plasma membrane is the outermost layer and it is made up of two components, right? It is made up of lipids and proteins. But in case you are asked to write an answer, what are the biomolecules that make it? And in case you get a diagram like this, I'm just drawing it. How does a plasma membrane looks like? So in case a diagram like this comes, right? So I'm drawing lipids right now. So this is one of the lipid layer. This is another lipid layer that I am drawing right now. So plasma membrane, it actually contains a lipid bilayer. And I hope you are clear that this is going to be the lipid bilayer. Okay, this is seen. And in case proteins are given to you, so this is the outside portion of the cell. This is the inside portion of the cell. Now, if proteins are there on the outer side, these are external proteins. Okay, because they are there on the outer side. In case you are seeing the proteins on the inner side, then these are known as internal proteins. So a diagram may come for interpretation about plasma membrane. So you must know that lipids and proteins should be visible. Now another type of proteins which are embedded here, right? So they are embedded here, right? So these are the transmembrane proteins. I hope it is clear now that they are inserted in the lipids, right? So these are also known as integral proteins. These will be studied by you in detail in case you will take bio in class 11th, right? So this diagram is of the plasma membrane. I'm writing it. This is plasma membrane, and this plasma membrane is made up of lipids and proteins. So here lipid is present as a bilayer. Bi means two. Layer means two layers are there. So one layer is this, another layer is this. Proteins on the outer, in the inner side, and some may cross the membrane. So this is how plasma membrane looks like. So we are done with the plasma membrane too. Now let's come to the another part. Now, if this is an animal cell, okay, you can make it of any shape. It's not required that it has to be circular only, okay? And this is the nuclear membrane that I am drawing. Whenever you are drawing the diagrams in biology, you don't require any ruler, okay? You can draw a 3D diagram. There will be no marks deduction. So I am making endoplasmic reticulum right now, okay? So this endoplasmic reticulum, because it is having ribosomes on its surface, which I am marking by blue dots, that's thus this endoplasmic reticulum is going to be named as rough endoplasmic reticulum. What is the function of rough endoplasmic reticulum? It helps in making the cytoplasmic framework, right? Similarly, you can make on this side as well. So in case you get a question on draw rough ER or SER, so because this side we don't have any ribosomes on its surface, so thus it is smooth endoplasmic reticulum. Okay, I hope you know the difference. Here, ribosomes will be absent. Virat Singh, this is of class 9. And here, in the case of rough endoplasmic reticulum, the ribosomes will be present. Okay, now you should also know what is the function of RER and SER. So RER helps in the synthesis of proteins because they have ribosomes on it. And SER because they are not having ribosomes. So one thing is understood that they will not help in the synthesis of protein. So their main job is in the synthesis of lipids, right? As we have just seen that plasma membrane is made up of lipids and proteins. So this endoplasmic reticulum, right? It helps in membrane biogenesis. Endoplasmic reticulum helps in membrane biogenesis. Very important question because membrane is made up of lipids and proteins. So lipids will be synthesized by SER and proteins will be synthesized by RER. So we are done with endoplasmic reticulum also. Till now, we are done with the plasma membrane. We are done with the endoplasmic reticulum. Let's come to the another part of the cell. Now, suppose again I have made animal cell. Okay. Now, in the center, of course, we will be having nucleus because this is an animal cell. So animal cell usually contains nucleus in the center. Endoplasmic reticulum we have already seen, but let's see if I want to compare endoplasmic reticulum with the Golgi body, then where is Golgi body located? Okay, so this green part is the ER. And let's draw Golgi body with the help of blue pen. So this is how we can draw Golgi body. So this is Golgi body or Golgi apparatus. Whatever name you want to write, you can write it, Golgi apparatus. Another thing, additional thing that I want to tell you is that Golgi body has got two phases. One is the cis phase, which is close to ER. Okay, the one end which is close to ER is cis phase, and the end which is far, it is known as trans phase. So this is far from ER. What is the job of, what is the role of Golgi apparatus? It helps in storage and packaging of the proteins which are formed by ER. It also helps in making the complex sugars from the simple ones, right? So Golgi apparatus is doing the work later when things are being synthesized by ER. Okay. So first, I think whenever you are making the Golgi body, also show ER because the teacher should know that you know the location of ER also. These circular structures which are there, they are known as vesicles. So with this, we are done with Golgi apparatus also. Now let's come to the another cell organelle. Again, in case you want to draw lysosomes. So there is no particular shape. You can draw of any shape. The membrane will be single, and lysosomes, they have hydrolytic enzymes inside them. So what is the meaning of hydrolytic enzymes? These hydrolytic enzymes will be doing hydrolysis, because of which carbohydrates, fats, and proteins may break down. So you can make lysosomes of any shape. Single membrane will be shown and hydrolytic enzymes will be shown inside. So we are done with lysosomes also. Now, let's come to the vacuoles. While making vacuoles, again, same thing, you can make them of any shape. And vacuoles, they help in osmoregulation. They can also help in storage. They can also help in excretion. And very important thing that vacuoles are lined by single membrane, and the membrane of vacuole is known as tonoplast. Okay. So vacuoles are single membranous organelle. The single membrane is known as tonoplast, and these are the three functions that you should remember. In case you are interested in hearing to the and understanding to the explanatory videos of fundamental unit of life, then you can come to the playlist of my channel. Uh, it will help you. T. Sai Swaroop. Ma'am, how to actually draw a heart? Dear, when I'll start with the live of class 10th, then I will help you how to understand where is each blood vessel. And there is no question in the past years where they have asked to draw the human heart. The only type of question that can be given to is you will be asked to label a particular blood vessel or a chamber of the heart. So you should know it thoroughly. Where is aorta? Where is inferior and superior vena cava? Where is pulmonary artery and vein? Where is left and right auricle ventricle? Right auricle and ventricle. So this way you should prepare for boards. Okay. So till now we are done with different types of cells that can be drawn. We are done with the plasma membrane. We are done with the endoplasmic reticulum. We are done with the Golgi apparatus. Now, suppose you want to show cell wall. Okay. In case you want to draw cell wall, then this is understood that cell wall is the outermost layer of the plant cell. Okay. So you can draw usually in a circular manner it is there. Okay. So you can draw it like this, and just to remark it, I am making dots here so that you can clearly see it. So this is cell wall, which is the outermost layer of a plant cell. In case you want to show plasma membrane, then we can make it like this. So this is plasma membrane. Plasma membrane. So this diagram is giving you a clear idea that cell wall is the outermost layer of the plant cell and plasma membrane occurs inner to the cell wall. But in animal cell, the outermost layer is the plasma membrane. So we are done with this as well. Now let's proceed and begin with mitochondria. For mitochondria, you should draw a long structure like this. Then, this is outer mitochondria membrane. Then for the inner one, you should draw finger projections like this. Okay, so this diagram is giving you an idea that this blue one is the inner mitochondrial inner mitochondrial membrane. This one is the outer mitochondrial membrane. So same mitochondrial membrane can be copied from here. These finger-like projections which you can see here are cristae. Okay. Now on these cristae we have small particles located on it. And what are they being named as? Oxysomes. Now, what is the role of oxysomes? I hope the screen is clear. Oxysomes, they basically help in ATP production. So, ATP production is due to them. Now, inside these inner mitochondrial membrane, I am making yellow dots, and these yellow dots signifies the matrix. Matrix means the cytoplasm of mitochondria. Very important thing is that matrix contains DNA and ribosomes. Okay, R for ribosomes. So this is the well-labeled diagram of mitochondria containing outer mitochondrial membrane, inner mitochondrial membrane. The surface area of inner mitochondrial membrane is more than the outer one. It has because it has finger-like projections called cristae. And on the cristae, oxysomes are located, which basically help in the ATP production. The cytoplasm of mitochondria is known as matrix, and matrix contain DNA and ribosomes both. Right? So we are done with the diagram of mitochondria also. Let's begin now with the diagram of chloroplast. So for chloroplast, you can draw an oval cell like this. This is the outer mitochondria membrane. I hope the screen is clear. Yes, now it is clear. So for drawing the chloroplast, we will be drawing the outer chloroplast membrane, then the inner chloroplast membrane. Just to differentiate, I'm drawing it with different colors, right? So this is outer chloroplast membrane. Whenever any membrane is written, that means it is made up of lipids and proteins. This inner one is the inner chloroplast membrane. So C for chloroplast, M for membrane. Now let's draw the inner part. So one coin, then another coin, then another coin, then another coin. That means we are making the pile of coins. So here I am making the structure of chloroplast, which is one of the type of plastid. So all these piles of coins, they are joined together. In case the diagram comes, then you should be able to draw this. So this one coin like structure, only a single coin, means I'm talking about this one. What can we call it? This is thylakoid. Okay? And this collection, all these coins together, what are they being known as? They are known as grana. 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 Okay? And the cytoplasm of the chloroplast. Cytoplasm of the chloroplast. This is known as stroma. Very important part. Stroma, the cytoplasm, and grana means the pile of coins. Stroma is the part that contains DNA and ribosomes, because of which chloroplast is known as a semi-autonomous cell organelle. So chloroplast is double membranous. Double membranous means it contains two membranes, outer chloroplast membrane and the inner chloroplast membrane. Now, single coin is known as thylakoid, and when collection of thylakoids is there, it is known as grana. All the granas are connected to each other by a structure which is known as lamella. It is not important for class 9, that is why I have not written. The cytoplasm of the chloroplast is known as stroma, which contains DNA and ribosomes. So till now we have done the diagram of plasma membrane, cell wall. We are done with mitochondria. We are done with ER. Now let's come to the control center of the cell. So the control center of the cell is the nucleus. Now suppose this is the plasma membrane, then we will be drawing the nucleus in the center. While drawing the nucleus, first please make a circle with this continuous lines like this, and thereafter join them like I am drawing. So please, from furthermore, do not make nucleus with two continuous circles. Why? Because there are nuclear pores also in the nucleus, which helps in the transfer of material between the nucleus and the cytoplasm. Okay, so what is this structure here? This is nucleus. Okay, and such a type of nucleus is actually seen in a eukaryotic cell. So we can call it as eukaryotic nucleus also, means it is well developed nucleus. This is outer nuclear membrane. And this on the inner side is inner nuclear membrane. N for nuclear, M for membrane. Now, in the nucleus, we have chromatin. So, what is this thread like material known as? Chromatin, Tin. Okay. Now, inside the nucleus, we have a dark stained body. This is known as nucleolus. Nucleolus. So, if you are able to see such type of diagram, please do correlate it with the eukaryotic nucleus or the well-developed nucleus, which is seen in eukaryotic cell. Now, this nucleus also contains the genetic material DNA. Now, in case a question comes, what is gene? So gene is a segment of DNA. Segment means a part of DNA. So don't get confused. If DNA is given to you, intact DNA is given to you, and a part of it we are talking about, then a part of that DNA is known as gene. Rahul, I am from Gurgaon. So this is the well-labeled diagram of a nucleus that can be seen in an eukaryotic cell. A eukaryotic cell is an advanced cell, highly developed cell. So till now, we are done with the diagrams of different types of cells seen in the human body. We are done with the diagram of plasma membrane, cell wall, endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, mitochondria, chloroplast, nucleus. Let's come to the diagram of ribosomes. In general, for a class 9 student, you know, ribosomes, they are made up of two subunits. One is the larger subunit and another one is the smaller subunit. Rahul, may I know from where are you watching this live? So you can simply draw a smaller subunit like this and a larger subunit like this, right? So blue one is the larger one and this one is the smaller one. So whenever these ribosomes they join, then only protein synthesis occurs. If these subunits are, so these both are the larger and smaller subunits. So in case both these subunits they join each other, then only protein synthesis will happen. Otherwise, protein synthesis will never happen. So with this, we are done with majority of the diagrams. Let me tell you a brief thing about the membrane composition of the cell organelles, like which one are amembranous. So the membrane is absent. Amembranous, A means absent. So once membrane is not there, it is amembranous. So ribosomes are amembranous. Now, in case a question comes that write the double membranous cell organelle. So we are writing this for cell organelles. So double membranous cell organelles are mitochondria and chloroplast. In case a question comes on single membranous cell organelles, the students who have joined the live now, in case you like it, please do hit the like button. And in case you are interested in hearing the explanatory videos, a playlist has already been uploaded. So endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, they are all single membranous. This part is very important. So this should be done with lot of revisions. We are done with the majority of the diagrams. Now let's see how a prokaryotic cell looks. So to draw a prokaryotic cell, again you will be making an oblong cell, and this is nucleoid, which is the poorly developed nucleus in the prokaryotic or the primitive cell. As you know that bacteria falls under the example of prokaryotic cell, so it has cilia on its surface. Cilia are the small hair-like projections, and this long hair-like projection, it is also seen in prokaryotic cell, which is known as flagella. So these small hair-like structures are cilia. And the only cell organelle which is found in the prokaryotic cell, it is ribosomes. So if a question comes, which cell organelle are common in prokaryotic and eukaryotic cell, then please choose ribosomes. So bacteria is an example of a prokaryotic cell. Pro means primitive, okay? And karyotic, karyotic means nucleus. So the cell with primitive type of nucleus is known as prokaryotic cell. And once evolution happened long way back, these were the cells that were formed. So in case it is asked which one is more primitive, answer prokaryotic. Which one is recent? Eukaryotic. Where is highly developed nucleus found? Eukaryotic. Okay. So with this, we are done how a prokaryotic cell looks. In case you want to make highly developed eukaryotic cell, then for making the eukaryotic cell, you can either make a plant cell or you can make an animal cell. So this is the plasma membrane that I have made. Now you can make nucleus in the center because I am making an animal cell, right? So this is nucleus with two double membrane. This is the chromatin. On this diagram, I will not do the labeling because I have done the labeling earlier. In case you have not seen the video at that time, you can replay the video. So this is green structure is chromatin. This red structure is nucleolus. And earlier I have not marked the cytoplasm of the nucleus. The cytoplasm of nucleus is known as nucleoplasm. What do we call it as? Nucleoplasm, right? So this is nucleoplasm, the black dots that I have just drawn. Now I am erasing it because I have to draw other cell organelles also. So this is the nucleus that I have made for you. Now let's make endoplasmic reticulum. So this is endoplasmic reticulum, and this is going to be SER. Now let's make RER. So RER will be the one that will have ribosomes on its surface. So this is RER, or rough endoplasmic reticulum. Now the membrane which shows cisterns means parallel tube-like structures arranged one after the other. This is known as Golgi apparatus. Rahul, no worries. I have just started recently with the live. However, I have already recorded videos. So whosoever will be here, I will be carrying on this live. In case you are interested, you can continue. So this is Golgi apparatus. In case it is animal cell, you can make less number of vacuoles. So these are vacuoles. Lysosomes can be made of any shape. They are single membranous, as I just spoke a few minutes back. So lysosomes, they are single membranous. They contain hydrolytic enzymes inside them. Now, the next one. In case you want to draw mitochondria, you can draw like this. Okay. So these are mitochondria. This is how you should draw mitochondria. Mitochondria is done, lysosomes done, vacuoles done. In case you want to draw ribosomes, because we do not show them joined to each other, you can simply show them with these green spots or black spots. It would not be an error. Okay. So these are all the parts you should definitely show in a eukaryotic cell. So the outer membrane is plasma membrane. This is mitochondria, lysosomes, ribosomes. Okay. Now, before I end, let's draw the components that are specifically seen in plant cell. So in case you are drawing a plant cell, make sure the outermost membrane is going to be the cell wall. So this is the plant cell, and we have cell wall as the outermost layer. So this is cell wall. I'm making it a bit dark so that it will become prominent. In case you have any assessment pending of the fundamental unit of life, you should also use pencil colors that enhances the visibility of each part. So this is cell wall. Now we have to draw plasma membrane. So this is plasma membrane. Right now I am drawing plant cell for you. That is why the outermost wall is this cell wall. This is plasma membrane. Now, as you know, that a large centrally placed vacuole is there in the plant cell. So this is a big vacuole that is seen. And the nucleus generally occurs in the periphery. Periphery means boundary. So this is the nucleus. And in case you want to show chloroplast, then chloroplast can be seen like this. The detailed diagram was already made few minutes back. In case you want to learn how to draw that, then you can learn by playing at that part. So in case they are asking you a question that draw a plant cell, then be careful that cell wall should definitely be there. Nucleus should be there at the periphery. Large central vacuole should be shown, and then chloroplast should also be shown. So we are done with the drawing of prokaryotic cell as well, eukaryotic cell as well, plant cell as well, and animal cell as well. In case this video was helpful to you, do consider subscribing. See you in next video. Thank you.